So if you recall from Arash's talk, he had some questions about, well, this is kind of part 2 to that, because he ended saying, well, I don't know if temperature affects the development of these changes in chemistry and symptoms in stored potatoes. Well, this talk here looks specifically at different storage temperatures, and we try to eliminate all the other mitigating factors that might be involved as well by basically taking potatoes and throwing them directly into cold storage temperatures. That being said, let me go ahead and walk you through the process of what we did and what we found, and we can go ahead and answer, well, partially Arash's question about, well, does temperature matter? Different temperatures matter. So, quick background, maybe if someone's tuning in online and doesn't know this stuff, yeah, zebra chip is a growing problem throughout the world caused by Lso, Candidatus Liberibacter, solanacearum, spread by potato psyllids. Hope all you guys know this. Basically the problem is it basically causes tubers to become unmarketable. And long-term sustainable management will ultimately be up to polytolerant and resistant cultivars. But most of my research and the stuff I do with other people generally relates around looking at tuber physiological changes and relating those to symptom development. What we found and published in the past is that when the fresh cut browning, enzymatic browning as they call it, is generally caused by higher phenolic levels, in particular chlorogenic acid, which I have a nice lovely picture here. That's actually the same exact compound that in apples, which is why I show this little cartoon of someone eating an apple. That causes the apple to turn brown after you bite into it and you put it down for a while and you come back to it later. Enzymatic browning process is caused by something called polyphenol oxidase, which is a protein that changes chlorogenic acid into a brown colored product. We also get increased browning when we fry them. That's a little different process. That's called non-enzymatic browning. That's generally caused, I hypothesize, by the Maillard reaction. It's a typical reaction in food chemistry that causes things turn brown when you fry them. Generally by cross-linking amino acid levels and reducing sugars, which is glucose and fructose. So just to give you guys a little more background on why these symptoms might be forming, there you go. So the problem that's going to be addressed in this talk is to carry on this idea that asymptomatic tubers can be accepted into commercial storage facilities, kept there for some time being, and we end up with these symptoms developing after they're removed from storage for final processing. So we wanted to look at the different aspects of that. Arash's study covered a lot of what happens with these late season infected tubers kept in storage, commercial storage conditions. So that was the first of 2 studies that we initiated a year ago to look at that. My study did something a little different. We wanted to see if there was a good storage temperature, if we can play around with the storage temperature, that final storage temperature, and whether that can affect the development of zebra chip symptoms in infected tubers, late season infected tubers. So what we ended up doing, what Charlie's lab ended up doing, is take 6 each red Lso and Russet Norkotah potato plants, inoculated with psyllid feeding, a central haplotype, as Charlie mentioned, a mixture of A and B Candidatus, and we also left 6 plants protected in these cages as controls. At harvest, they have lovely pictures showing you how they took the plugs at the stolen end of the tubers, and then they were placed immediately into cold storage at 3, 6, or 9 degrees Celsius. Being a scientist, I go to Celsius, but that's 37, 43, or 48 degrees Fahrenheit. So the 43 is pretty close to the 45 I believe Arash used. 48 is a little higher. 37 is quite a bit lower. The plugs were collected, we took additional plugs 2, 4, 8, and 12 weeks after being placed in that cold storage. All the plugs were analyzed for Lso. Amino acid, sugars, or phenolics. And at 12 weeks, at the very end of the study, I got slices so I could actually look at fresh and fried symptoms and get those ratings too. Or maybe Lee did that. I forget who exactly did that. But we have fresh and fried symptoms at the end of the study. So a quick process, we get the samples, we weigh them out after mashing them down a little bit. And then we extract in either buffer or methanol. The buffer, just a simple phosphate buffered saline, eye wash solution more or less. We can use it in a commercial kit to look at amino acids on gas chromatography, which is that machine at the top there. We can also put the buffer directly into HPLC, equipped with something called an ion exchange column to separate out sugars, so a little more accurate than bricks perhaps. And then we take the methanol extracts and we can use those into a different HPLC with a reverse phase column, as they call it, to look at phenolics. I also have an MS detector with the HPLC to help me identify what some of the phenol compounds are. So the results. So I wouldn't say these are the most thrilling results in ever that I looked at, but the results. So what we find is that in terms of fresh and fried symptoms for red Lso Titer, I'm going to take each cultivar at a time. So with red Lso Titer, we had much higher symptoms with the tubers placed at 3 degrees than at 6 or 9. Although the fried 9s weren't really statistically significant in the 3s. Fried symptoms got up to about a 2 rating, which is pretty moderate. Fresh symptoms were just a little over 1. So we did have some development of symptoms after these things were placed in storage. Based on red Lso Titer, seems like 6 degrees, which is about 44 degrees Fahrenheit, was probably preferable in terms of keeping symptoms down. In terms of Russet Norkotahs, we kind of see similar results. Statistically, though, there was no difference, even though there looks like there might be a difference here. But statistically, there was no difference due to temperature and fried symptoms for russets, but there was for fresh symptoms with much lower levels for the tubers kept at 6 or 9 versus the tubers kept at 3 degrees Celsius. In terms of Lso titers, this is actually kind of probably the most surprising thing and complicated thing for me. These are relative LSO quantification that we did. So higher numbers should be higher levels of bacteria, higher titers. And if you look at the 3s for this red Lso Titer data, regardless of week, they're all pretty low. And although we have incredible variance tuber to tuber, so none of these actually were really statistically significant, which due to this incredibly variation between the tubers. The other thing that I thought was a little surprising is, high numbers up here, lower numbers down here. You would think the numbers would have gone up over time. And we really didn't get that, although I don't think there was a significant weak effect because once again we had a lot of variation tuber to tuber. So there's probably going to need to go back and to still look at the data again to really maybe even rerun some of these titer data to really try to eliminate some of this variation and figure out there is a problem with the PCR or something. Maybe to tighten that up, but this could be a real result. We could have very variable data in terms of the development of LSO titers and stored tubers. We actually got a very similar situation, somewhat similar situation with Russet Norkotah as well. Quite a bit of variation. So once again, there's no letters here because there's no significant differences. Even though this one's way up here, but the error bar goes down to there. So, too much variation to really statistically say these things are significant. But at least in this case, you kind of had an overall trend, especially the 9s, that the levels went up over time, maybe with the greens as well. Once again, even though we have the most symptoms at 3 degrees Celsius, very, very low titers throughout the whole study. So I don't know what to make of that. It's kind of surprising, almost conflicting data, but it is what it is. I did run the chemistry. So these are actually me adding together all individual amino acid levels together, micromoles per gram fresh weight. And what we end up finding here is that for both red Lso Titer and Russet Norkotah, that overall the affected, also inoculated tubers had higher levels of these amino acids than controls. But there was really pretty much the levels of controls and LSOs for 6 or 9 are basically right on top of each other, all here and here. Although for some reason the controls 6 are always lower throughout the whole study for Russet and Norkotah. So this kind of shows you that there might be a little bit of difference going on at 3 degrees between infecteds and controls, but for the most part, it doesn't seem to really matter in terms of changes of amino acids at these temperatures, at 6 or 9 especially. The other thing is the levels of these things are generally flat, there and there. So they really didn't change much at all once these were kept in these storage temperatures. In terms of total sugars, basically I added together the glucose and fructose levels to kind of simplify things a bit. Once again, levels are generally flat. You can especially see it with the Russet Norkotah. I don't know why that's way up there, the LSO at 3 degrees, but after this time period, you know, everything's right on top of each other, similar levels, no statistical significance. With regard to the soda, we see higher levels, though, at the 3 degree samples and controls versus LSO inoculated. And the same with controls and LSO inoculated for at 6 degrees. So in that case, it seems that it actually might be higher levels in controls. If you remember Rashed's talk, he actually had this negative correlation with sugar levels and LSO titers. Well, I don't know, this might be somewhat a real result because of that or not. Once again, quite a bit of variation, so it's hard to say statistically if anything really went on. In terms of chlorogenic acid, the one phenolic compounds that probably lead to this brown color when freshly cut. Again, we have higher levels at 3 degrees. It doesn't really seem to matter too much if they're affected or non-affected. The levels at 3 degrees, they go up over time. The others kind of increase somewhat as well, regardless of infection status or inoculation status, but there's really no significant differences between those other temperatures, except for this control at 6 degrees, which jumped way up there for 12 weeks. In terms of flavonoids, which is the other major compound class of phenolic compound you can find in potatoes, once again, general increase over time after being in storage, higher levels at 3 degrees for Russet Norkotah here, not so much though for the soda here. I don't know what that means. A lot of variability, which makes conclusions really hard to reach. So I don't know how I am on time, but some generic, oh good. So some generic conclusions. I think we had plenty of samples for this. So, you know, this variability, I think, is just due to the way the study was conducted and maybe the way that these potatoes naturally behave in storage. Because we had a total of 300 samples. So we had 6 plants, but we took all the tubers from all those 6 plants. So we had a lot of tubers placed in storage. We also had 18 amino acids we looked at, fructose, glucose, 6 chlorogenic acids, 5 flavonoids. That was a total of an Excel file of like 25,000 data points. So a lot of statistics for me to run through, and I should have enough power to really wiggle out if there's anything really there. And, you know, but there's a lot of variability in the study still regardless. The symptoms are, well, long story short, the symptoms levels were greater at the tubers kept at 3 degrees and 6 or 9. So it does seem that you probably want to not store your tubers too much below 6 degrees, about 45 degrees Fahrenheit, 43 to 45 degrees Fahrenheit, based on the symptom data we have. But strangely enough, those LSO titers, they were really low at the 3 degrees. So I don't know if it could just be that the higher symptoms we saw at the 3 degree temperature is just something that happens in potatoes kept too cold versus something that the bacteria is actually doing. So I don't know if anyone has any thoughts about that. Either that or just you read around the PCRs. The other thing about the study too is we took plugs at the top of the potatoes. So, you know, this variability issue, and especially this titer matching some of this other stuff, could just apply that maybe these the distribution of bacteria and the changes in physiology could be more localized than we thought before. So all the prior studies I ran, we did slices, and I basically took a lot more material through that slice, the vascular bundles, all the way around in a big ring, and mixed it all together, and then took subsamples out of that for analysis. So basically, I homogenized the tuber samples a little bit. In this case, we took very small, little tiny samples of very specific spots around the top of the potato tubers, because we had to use the same tubers repeatedly measured over time. And so this could imply that actually we were just kind of hitting hot and cold spots as we were doing that over time, leading to some of these results. So it is what it is, but it might warrant further investigation to actually look very specifically to see how uniform LSO might be distributed in potatoes, as well as how uniform the physiological changes in response to LSO might actually be. I don't know, just throwing that out there. And here's the conclusions from the chemistry. Bearing in mind what I just said, we did see higher amino acid levels at 3 degrees, and LSO infected or treated tubers than controls. Other temperatures were fairly similar. Reducing sugar levels were generally greater in controls, especially with red Lso potatoes. Chlorogenic acids were higher at 3 degrees, tubers in 3 degrees, especially after 12 weeks, than others. Russet flavonoid levels kind of were all over the place, but they were higher at 3 degrees than other temperatures. And it seems based mainly on the symptom data that keeping the tubers in long-term cold storage at around 6 degrees C is probably the way to go. Not go too lower than that or too much higher than that. So I'd like to acknowledge some people who helped me out here. So these are my lab members. Noemi Fonseca, in particularly, she went from picking crops in the field to my lab to basically put her way through college. And she basically processed, over the last winter, probably close to like 2,000 samples for me, this project and others. So very hard worker. She really seems like she wants to stick with science, which is great for her. I just wanted to point her out. The funding was provided by the SCRI mini grant program from a couple years ago, as well as base funds from USDA.